Okay, so here's the pancreas. It consists of right and left lobes that join to form a small central body. The left lobe, contained in the dorsal leaf of the greater omentum, begins at the pylorus and extends along the greater curvature of the stomach. The right lobe is contained within the mesoduodenum and is closely associated with, well, as you might expect, the duodenum. Imagine the pancreas as someone leading a split life, for it is a dual-functioning, glandular organ that serves two incredibly different and vital purposes for the body. To get to the point, the pancreas contains both exocrine and endocrine glands, making it a key player in both digestion and metabolism of nutrients. The exocrine pancreas makes up about 98% of the pancreatic tissue and is responsible for GI function, primarily the synthesis, storage, and release of enzymes involved in the digestion of carbohydrates, proteins, and fats. But for the purposes of this video, we don't really care about the exocrine pancreas. That's because the subject of this video is the endocrine pancreas. The endocrine pancreas, making up about 2% of the pancreatic tissue, is organized as discrete islets, or islands, known as the islets of Langerhans. The islets contain 4 cell types, each of which produce a different hormone. Alpha cells produce glucagon. Beta cells produce insulin. Delta cells produce somatostatin. And PP or F cells produce pancreatic polypeptide. For this video, we're going to focus only on alpha and beta cells. But just know that all of these hormones are involved in controlling the body's metabolism, and more specifically, in glucose homeostasis. We'll start with beta cells. The primary function of a beta cell is the storage and release of insulin. So what is insulin?
Well, technically speaking, insulin is a protein consisting of 2 chains. The monomer form of the hormone
is thought to be the active form. Insulin also exists in dimer and hexamer forms.
The latter complexed with 2 zinc molecules.
Alright, let's cut to the good stuff, shall we? Insulin is an anabolic hormone with the net effect of lowering the blood's concentrations of glucose, amino acids, and fatty acids, and promoting intracellular conversion of these compounds to their storage forms, glycogen, protein, and triglycerides. You see, without insulin, glucose does not easily penetrate cell membranes, except in a few tissues, such as the brain and the liver. But for most other tissues, you can think of insulin acting sort of like a key, unlocking doors to cells and allowing them to take in necessary nutrients to be used as an energy resource. In short, the presence of insulin is critical to the movement of glucose out of the blood through the plasma membrane and into the cell. So, as the blood concentration of glucose rises, such as after breakfast, or after lunch, or dinner, or even after a healthy snack like, a banana. Or if you're a dog, then, well, dog food. Insulin is synthesized and released by the beta cells of the pancreatic islets to bring blood glucose into cells and back to an acceptable blood concentration. So what about alpha cells? Alpha cells are responsible for the production of glucagon, which also has a major role in maintaining normal concentrations of glucose in the blood. Glucagon is a catabolic hormone and is often described as having an opposite effect of insulin. Simply put, the pancreas releases glucagon when the blood sugar concentration levels fall too low, such as in the period of time between breakfast and lunch, or between lunch and your late afternoon banana. In fact, it's even secreted to help balance insulin during meal times, to help prevent hypoglycemia after glucose has left the blood to be stored or used as energy in cells. Glucagon targets hepatocytes in the liver, where its major effect is to increase glycogenolysis, decrease glycogen synthesis, and increase gluconeogenesis. The net result is an increase in glucose concentrations in the blood during states of hypoglycemia, like, for example, when you're super hungry. I like to think of insulin and glucagon as being in a very healthy, egalitarian relationship, perfectly counterbalancing the effects of one another in association with the daily ingestion of food and the intervals between food intake to maintain glucose homeostasis. So what happens when the balanced relationship between insulin and glucagon becomes not so healthy? What if insulin came home from work one night and caught glucagon and another hormone doing the horizontal hokey pokey and then decided to pack up and move out? Well, people, this is how the pathophysiology of diabetes begins. To learn more about the pathophysiology of diabetes mellitus, watch part 2 of this series.